This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 256, recorded on October 24th, 2013. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiV, the podcast all about viruses. Joining me today here in the TWiV studio, Dickson Despommier.
Good morning, Vincent.
It is a morning, unusual recording hour.
Indeed.
You can have your morning voice.
Well then, looking out the window.
It's nice out, isn't it?
It's gorgeous out here.
But
it's a beautiful fall day.
It's like 8 degrees Celsius.
Yeah, it's a little cool, but you know what?
No, not a cloud in the sky. Beautiful.
It's gorgeous.
It's only going up to 12 here in New York City.
Yep.
Let's find out what the weather is throughout the rest of the United States.
You know
what do you think?
This will be good.
Joining us today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. It's 2 degrees Celsius,
Wow.
36 Fahrenheit.
Got some frost on the ground?
There was some frost on the cars that I noticed, and yesterday I think was colder.
Wow. Yeah, I guess fall's here.
Mm-hmm.
Oh, yeah.
Let's go down to Florida, north central Florida, and see how the weather is there, where we'll find Rich Condit.
Hi, everybody. How you doing?
We're okay. You have
we've got 53 degrees Fahrenheit, 12 Celsius,
a little chilly.
which is chilly for Florida.
True.
I would say mostly high wispy clouds, 63% humidity, dew point 41 degrees, so it's dry as well.
Nice.
Right.
But that's it for hot weather down there, right?
Well, this is a sort of unseasonably cold by, maybe 5 degrees, maybe even 10. I looked at the high today is forecast to be 75. Later in the week, it's going to go up to the low 80s, something like that. That's more typical. Okay. So I had to dress up a little bit for my ride in this morning, but it was brisk and refreshing.
By your ride, you mean your bike ride?
Yeah.
Yeah.
Great.
Not my horse ride. Okay.
Let's see what the weather is in western Massachusetts, where Alan Dove can be found.
That's a great idea.
Here I can be found, yes. It's 46 Fahrenheit, 8 Celsius. Nice brisk fall day. The leaves are turning.
Yeah, finally.
Solar flux index is 156, and the A is 4 and the K is 1. So
we have new metrics.
The sunspots are pretty good today.
Oh, good. That's about time they came back.
Yeah.
Well, as you see, we have a full TWiV crew.
Full boat.
We're full of TWiV.
Full boat.
Full boat. So
right.
we have papers today to do, but before we get to that, a couple of things. First of all, today is World Polio Day.
Uh-huh.
Hooray.
Celebrate how far we've come in cutting down the number of polio cases.
Still work to do. We're still playing whack-a-mole.
We got some outbreak in Syria recently, right?
Yeah.
That's totally expected.
And that is because the immunization rate has gone way down.
Absolutely.
So, yeah, whack-a-mole. It's really hard when this happens because then you get circulation of wild viruses. And in Israel, they're in the sewer, even though they're not causing polio, but you can see what happens. So it's a tough one.
But progress has been made.
A lot of progress from the 1980s, where there were at least 350,000 reported cases a year globally.
Yeah.
We have a few hundred a year now. So it's pretty good.
This link that you've picked for World Polio Day is actually quite good.
This is Kathy, right?
I put that in.
I could
tell
by the green font.
Yes. Oh, but I didn't do them all day.
I did them all day. So yesterday was Mole Day, October 23rd. 10 to the 23, Dickson, you get it?
Yeah, I know. No, you explained that earlier to me, and I actually did get it.
Usually celebrated at 6:02 in the morning.
It's 6:02.
So my son this morning, my two kids celebrated it in chemistry this week. They made Mole Day T-shirts, And my son said, why don't we celebrate it on June 2nd? Which could be, right?
Yeah.
But 10:20, but you're right. If you celebrate it at 6:02, everyone knows what we're talking about, I hope.
Oh, I hope so.
If you don't, send us an email.
Right.
Check out Avogadro's
Check out the link.
Right. And also, sadly, a few weeks ago, John Holland passed away, someone we've mentioned often and who you worked with, Kathy, right?
Yes. I and a lot of other people counted up somewhere on the order of 57 people from an email that he sent me once listing everybody. At least 21 people in academic faculty positions, including Luis Villarreal, who was on TWiV 24, and Bert Semler on TWiV 97. And at least 5 or 6 people I can think of in industry, including Ed Penhote, who was the co-founder of Chiron.
Right.
So lots of people out there in academia trained with him. He's just a really great mentor. I was listening to the TWiV from last week, and just like Chris described Jim Pippis, being in Holland's lab was crazy good. And he was a mentor by example. He worked at the lab bench despite all his other responsibilities, and so people have been sharing different comments about him. One person said, being side by side with him in the lab was both scientifically stimulating and entertaining. He made science exciting and will be missed. Here's another one. By all the modern theories of proper scientific mentorship, parentheses, weekly documented meetings with progress reports and specific aims achieved on schedule, he was a terrible mentor. Ha! But what a great mentor he was. Goes to show the power of his intellect coupled with the joy, excitement, imagination, and enthusiasm that he shared with us every day. And as some other person said, his personal engagement and informal style served as better mentoring than any lab meetings or committees could offer.
That's really outstanding. I really love to hear that.
Yeah.
That's great. Because I'm completely disorganized.
Yeah.
I don't do any of this stuff that you're supposed to do. Jeez. I met John just once, and I believe 1976 when I was visiting California and kind of poking around while I was stationed in England. I made a trip, and as I recall, my conversation with him had to be terminated because he had to take down a centrifuge. He had a spin going.
Interesting.
Yep.
Dickson, you ever meet him?
Nope.
I never did, but
Where was he based?
Well, mostly at UC San Diego from around 1968 on. But he was the founding chair at UC Irvine at age 35 or so in 1964.
Wow.
Yeah.
So I gave a seminar early in my career here in the 80s at Scripps. And back then we were working on polio receptor, and I used to cite a lot of his work from the 60s where they were interested in tropism and the relationship to receptors, and he used polio as a model. And in my talk, I mentioned John Holland thought this and this and this and this.
Yeah.
All right, so I finished my talk, and then I'm talking with, I think, Mike Oldstone. He said, John Holland was in your audience, and he really liked that you mentioned him. I said, I didn't see him. He was in the back, and I remember the tall guy was standing in the back of the room, kind of smiling, and then he just left.
Yeah.
Had another centrifuge to take.
Yeah, probably. So that was my only in vivo experience with John Holland, but it was great.
I'm
liked
sorry
him.
I never did meet him.
Yeah. You have something else in common with him besides working on polio. Like you, he won the Eli Lilly Award. And like you, his postdoc mentor had won the Eli Lilly Award.
Wow.
So kind of a nice...
Who was his postdoc mentor?
Jerry Siverton at University of Minnesota.
Right. Yeah, those papers I used to cite all the time.
Yeah.
I never met him, but I certainly read a bunch of his old papers when I was in Vincent's lab.
One of the papers you put here has a great picture of a polio plaque assay in glass cell culture bottles.
Yeah.
I love it.
Yeah, it shows them with caps on, but by the time I got to the lab, the caps weren't holding the CO2 anymore, so we used rubber stoppers, which led to a stopper contest game thing in the lab. But
so you used to fill them with CO2 and then cap them, right? Because we didn't have CO2 incubators.
Yeah. In fact, we did all our cell culture on the bench.
Yeah,
we didn't have hoods.
Yeah, me too. I did that even in Peter Palazzi's lab. We had a room where you just did it out on the bench. It was fine. It worked.
Yeah, ours wasn't even a special room. Ours was the main lab.
The main lab, yeah.
Try writing that up in the methods section. I want to see those papers.
Well, and I remember when I first joined the lab, which was 3.5 years into my graduate career, I had rotated in a lab that used tissue culture hoods, but but John's lab didn't have those. And the Frank Hordisky next to me on the bench said, "Yeah, Kathy, sometimes when I'm passing my cells here on the bench, I just imagine that there's a big hood over me." So doing that, seemed to work.
Things have changed.
Things have changed.
Yeah, but he kept working in the lab. He retired in '95, but he had a bunch of papers after that, and really, in addition to the work on polio, then did things on vesicular stomatitis virus and also rabies, particularly persistent infections and DI particles, and then made the move into understanding RNA virus evolution. Did a lot of work there. I don't know if he was the founder of the quasi-species theory, but he certainly was a strong proponent of it and provided a lot of evidence for those kinds of things. So, yeah. All
right. I'm sorry we never had him on TWiV also.
No, and I was thinking of that just a couple of weeks ago when we were talking about we need to get these people out. And then I'm not sure if he would have done it.
Yeah,
he might have, but he might have just said, "Nah, I won't do that."
Yeah.
But whatever.
Yep, I've had that happen a few times. No, I don't want to do it.
Yeah.
Yep, I agree.
All right. That's John Holland. And we have two papers that were mentioned in last week's TWiV in Austin. If you should go back, listeners, and listen to that one because Chris Sullivan gave a great introduction to RNA interference.
He really did a nice job summarizing that whole thing. It's pretty intense. You have to listen carefully, but it's all there and it's very coherent.
Yeah, I've listened to that part of it twice already, and I've listened to the whole TWiV once. It's a really outstanding TWiV because the second half with Krug is also good. So
yes,
yeah, I like Bob's too because he went back in history and
I like the backstory on the cap snatching thing.
It's good to have that. So Chris mentioned two papers that had recently been published in Science, which he had suggested demonstrated antiviral RNA interference in mammalian cells. So I thought we should do those today. And just to remind everyone that RNA interference is something that is present in plants and insects and nematodes. It is also present in mammalian cells, but whether it's antiviral in mammalian cells has been controversial.
Right. So this is a system that produces short pieces of RNA that bind transcripts and target them for destruction to silence them.
Right.
So, one of the things that I've found a little confusing is the terminology here because we've got interfering RNAs, we've got siRNAs, we've got microRNAs, we've got all kinds of stuff. And I talked to Chris. I talked to Chris after his TWiV, and basically he uses RNAi or interfering RNA as sort of an umbrella term to cover the whole thing. To cover the whole idea that these small RNAs can have regulatory roles. And under that umbrella are included things like microRNAs, that have a very specific different function.
Yeah,
and that cleared up a lot of stuff for me.
The terminology in this field gets confusing partly because a lot of the boundaries between different RNA species are quite arbitrary.
Mm-hmm.
Yeah, I think,
if it's this length, then it's siRNA. If it's this length, then it's miRNA, and then there are the long non-coding RNAs that are over 200 bases, and there's this whole universe of RNA-mediated gene regulation that goes on in cells.
I think,
and some people make a distinction if you transfect in synthetic short RNAs, and some people say that that's just colloquial to refer to them as siRNAs. That was mentioned in this cell-hosted microbe mini review.
Right.
So, yeah.
But everybody does it.
Right.
But that shows that mammalian cells have the RISC machinery for targeting mRNAs, right?
Right. The machinery for doing this, right, is present in all of these different in I guess all eukaryotic cells, right?
Yeah,
I think so.
Yeah, and it appears to be a normal part of gene regulation machinery that you routinely transcribe genes and then destroy the transcripts in this horribly inefficient manner to inhibit production of the protein. So there's this whole gene regulation network that's going on with it. But the question was, well, in plants, this is also an antiviral system that that attacks viruses that are invading the cell, but nobody was really sure if that happened in mammals.
Why did it take so long then?
Because
you have other interference.
You'll see it, I think, as we go through the papers. It's not clear. I think it's not yet clear, okay? So when I heard Chris talk about these papers last week, it sounded like this was the answer, but there is actually a little mini review in Cell Host and Microbe which points out the shortcomings of these papers. So we'll get into that.
So interfering RNAs in the context of viral interference would be a virus infects a cell, and some of the during the infection, some of the RNA that gets processed by this specialized machinery down into these short RNAs, which then can target the viral RNA for degradation or suppression, translation, right? And that is known to occur in what? Insects
and plants. Right.
And the machinery is present in mammalian cells, but it wasn't clear that it could be used for RNA interference.
Right.
That's the big deal.
And so you may ask, how do viruses of plants and insects replicate? In the face of that, well, many of them encode viral suppressors of RNAi, which are VSRs, right?
And when Vince told me that, I said, aha, that's another example of spy versus spy.
Yep.
Yeah, of course.
So these are old stories told in a different way.
So the first paper is by Li, Lu, Han, Fan, and Ding. It's called RNA Interference Functions as an Antiviral Immunity Mechanism in Mammals, which I think, having read the paper, is a little bit of a strong title, but we'll let you decide. In this paper, they use nodavirus.
They're from Irvine, by the way.
Riverside.
Riverside.
Oh, I'm sorry. Riverside?
It is UC.
UC Riverside.
Yep. Right. And nodavirus, which is an interesting virus that can replicate both in mammals and in insects. All right?
That's not unusual.
Tell me another one.
Yellow fever.
Another one.
I think
West
a
Nile.
lot of
I can go on. All of the arboviruses seem to be able to do that.
Arboviruses replicate,
so they're going to call this one an arbovirus too, simply by definition, because it's transmitted by an arbo type thing.
Hold that thought.
Okay.
Something that has to go in insects, okay? Because you might predict that it needs a viral suppressor of RNAi. And then if it goes into a mammal, it may or may not need that, depending on whether there's antiviral RNAi in mammals.
Interesting. I hadn't thought about it that way.
Multiple hosts.
So their logic here is they say, nobody's found antiviral RNAi in mammals. Maybe that's because, the viruses all encode suppressors. So what if we took a suppressor out? So in nodavirus, the suppressor has been identified. It's a protein called B2.
Okay.
So they use wild-type nodavirus and a deletion lacking the B2 gene. Right, that's their approach. Mm-hmm. And so they infect baby hamster kidney cells with this virus, and they look for small RNAs. You can extract small RNAs and sequence them and see how long they are and where on the genome they map. And when they use wild-type virus, they find small RNAs, but around the canonical siRNAs are 22 bases long. They're duplexes with 3' 2 base overhangs, right?
Right.
Talk about your jargon.
Well, that's molecular biology.
Re-examine that statement. I feel so sorry for those pharmacologists out there listening to this, and we're trying to derive something from our conversation.
Well, there's a specific
Picture a double-stranded nucleic acid.
No, I know what it is.
Can you do that? And you got a little bit of nucleic acid hanging off the end.
Thank you so much.
Right.
No, you mentioned canonical.
Canonical.
You mentioned the word canonical without
canonical.
That
it has a certain structure, and that's considered to be. And if you look at the gene, so these are processed from the double-stranded intermediates in viral replication, and they also use another phrase. These siRNAs are phased. So, dicer is going down the genome and chopping as it goes along. So it's going to make overlapping, right? Mm-hmm. Phased.
Got it.
Tiling.
Mechanism. Tiling, that's right.
So when they infect with the wild-type virus, they see a lot of small RNAs, mostly from the viral plus strand, and that's not right for a canonical siRNA.
And they figure that's just degradation.
Which I was really, that's a lot of degradation, right? Although the percentage is not so high, I think. It's
it's a mess in there.
It's a mess. It always has been a mess.
Right.
So they use the mutant virus lacking the suppressor B2, and then they see canonical siRNAs from both strands. You see plus and minus strand, which is what you would expect. And they go through some interesting analysis to show that they are canonical. So you take away the suppressor, and you get what look like 22 nucleotide interfering RNAs. Right? Now, how about viral replication?
How about that?
So if you take out, this is viral replication in BHK cells, they do a kind of growth curve, but they're measuring RNA. I would have liked to see a plaque assay, but nobody does plaque assays.
We're just old-fashioned, Vincent.
Does this virus plaque?
I'm sure it does.
Okay.
I'm sure it does. It's a beautiful virus. And you get a nice accumulation.
But I thought it was not a virus.
It's not a virus.
It's a protozoan.
Anyway, you get growth, a nice single-step growth curve. You get 10 to the 3. I don't know what that means in terms of RNA. RNA accumulation folds, Just give me PFU. But when you take out the B2 gene, it goes down 1 to 2 logs, I would say. So there's a defect in replication associated with making these 22 nucleotide RNAs. Okay? You can also see by northern blot that, the viral RNAs replicate very well in wild-type virus-infected cells. But if you take out this B2 protein, you don't get a lot of RNA accumulation.
And if you put the B2 protein back into the cells,
Right.
then you rescue the replication. So it really is the B2.
It's the
that's doing
B2
this.
deletion that's doing that. They also put it in a VP35 gene from Ebola virus, which has been suggested to be a viral suppressor of RNA.
Right.
It supposedly interacts with some of the proteins that the dicer enzyme needs, and that also rescues the RNA synthesis.
Now I was a little confused about this because one of the arguments is that VP35 suppresses the antiviral response by a different mechanism than double-stranded RNA binding, but by binding the machinery or interacting with the machinery that processes the RNA, which to me is an important point. But is that true? Because does VP35 have double-stranded RNA binding properties?
According to this review article, yes. Okay. It says, of note, BP35 is in fact a double-stranded RNA binding protein that has been shown to also block the interferon response.
Okay.
So
it's not straightforward there.
I don't think so. I mean, so far, these are associations, right?
Right.
They also do a point mutant of B2, a single amino acid change, has the same phenotype, to get around arguments that you're really mucking up things. And you also get these siRNAs. They do some sequencing to show what the siRNAs are. And, then they infect mice with these viruses. So let's before we move into mice. So far we have some correlation in cell culture, but my understanding is from the field that what you'd really like is to show that this is dicer or Argonaut dependent, right?
I would think so, yeah.
In fact, that's what this review says, that you have to show that, and they haven't done that in this. And of course, I think it's not, I don't know the field really well, but I think it's really hard to do knockdowns of dicer, right, in mammalian cells, because then you mess up all the microRNA machinery. Is that correct?
I would think you would get, I can't state you chapter and verse, but it wouldn't surprise me at all if there were significant pleiotropic effects.
Well, we're going to get to that in the next paper, aren't we?
Yeah, I mean,
well,
that's
going to be done in different kinds of cells. Remember, these are somatic cells.
Right.
So the question is, in baby hamster kidney, I would think they would have done it if they could.
Yeah,
it's glaringly absent. And so what I have here, and I think is just a correlation between having the right siRNAs and not replicating well. And it happens to correlate with the presence or absence of this B2 protein, but could be.
There are dicer knockout mice, and you can make dicer,
right.
I would assume, dicer knockout cells from that, but I forget the details of their phenotypes.
Okay. So they infect mice with with these viruses. The wild-type virus is lethal and replicates like gangbusters in mice. They put it in the foot pad, I think, and they look in the hind limbs and forelimbs for the virus, and it spreads. And the mutant, lacking B2, is attenuated. The mice survive. They even have a picture of surviving mice.
Yes.
Although I don't know.
I don't know what the picture shows.
Yeah, I don't know why you have a picture of surviving. It doesn't tell you anything. It's really strange.
It's a color picture, so they probably had to pay for it. Who knows? Yeah.
And in mice, so the mutant doesn't accumulate its RNAs very well, and you make lots of siRNAs that you can detect on a northern blot, actually. So the mutant virus in mice induces lots of the right siRNAs, and they sequence those and they say they're canonical, et cetera, et cetera.
Excuse me, just looked it up here. Loss of Dicer-1 led to lethality in early development with Dicer-1 null embryos depleted of stem cells. So it's an embryonic lethal. It's a serious protein.
Makes it hard to work with those mice.
It does,
Yeah.
doesn't it?
So
It makes it dicey.
that's basically the result. And now let me tell you what they say in this paper. They say that the data show that the ability of these nodavirus mutants to express a functional viral double-stranded RNA binding protein is critical to their pathogenesis, rather than definitively establishing that siRNAs are antiviral and these proteins act through antagonisms of such siRNAs. So I guess they would like to show in both of these experiments, in cell culture and in mice, that the siRNAs are actually antiviral, that they actually can target the viral genome and chop it up.
Right. What they've shown is that the gene in the virus that silences the siRNA production is necessary for the virus to go through its normal replication cycle.
Right.
And that there's a correlation between that and whether or not these things that look like siRNAs show up.
Right. There's a solid correlation between that and the production of siRNAs, which is really, really close to showing that this is an essential system for the virus, for this infection, and that this is an essential and normal antimicrobial response in the cells, but it's not quite there.
And that's why I think the title is really strong, because they say RNA interference functions as an antiviral immunity mechanism in mammals. I think that based on the data, that's really strong. And this review, which I highly recommend reading, although it's unfortunately in Cell Host and Microbe, it is by three of the players in the RNA interference field: Brian Cullen, Sarah Cherry, and Ben Tenover. It's really good. So these are people who work in it and know a lot more than I do, so check it out.
There's also a review in the Science by Selena Sagan and Peter Sarnow that has a nice picture describing the mouse experiments with the suckling mice versus adult mice that I kept referring to as I read it.
Right. And that is not it's not critical as you would expect, because it's in Science, which is where the papers are.
No, that's what I was going to say.
But it is an excellent summary. Unfortunately, it is also, and so are these papers, paywalled.
Yeah, that's really sad.
I just ripped them out and brought them in.
Yeah, well, you buy the journal.
I do. I'm
sorry, but anyway, people can read them vicariously through TWiV, right?
We could.
Alan, did you just verb the word paywall?
They're paywalled.
Yes, I'm sorry.
It's okay. I can deal with it. I just wanted to make sure.
Well, you can wall things.
Yeah. Okay. Why not paywall?
Why not paywalled?
Okay.
Yep.
Is that wall concrete or plaster?
Mr. Gorbachev, tear down this wall. No.
All right, the second paper in Science is called Antiviral RNA Interference in Mammalian Cells.
Right.
By Maillard, Chiaudo, Marche, Lee J, Ding, and Voinet.
Now, this Lee is the same Lee as on the other paper, right?
Lee and Ding both. Yes.
Lee and Ding, right.
Little collaboration there.
And the other investigators are from Switzerland, Swiss Federal Institute of Technology in Zurich.
Mm-hmm. And the collaborators from Riverside. So in this paper, they take a slightly different approach, although there is some overlap, as you will see. There is some evidence that in embryonic stem cells, that the RNA interference machinery can deal with long double-stranded RNAs, okay, as opposed to somatic cells where it's been difficult to show that a viral double-stranded RNA can be chopped up. Here, apparently, there's some evidence that it can happen. So they culture mouse embryonic stem cells and infect them with encephalomyocarditis virus, EMCV, which is a picornavirus, and which we happen to work on. All right? So you infect these ES cells with EMCV, and then they look for siRNAs, and they find them.
In terms
Yes.
of virology, there's a gross similarity between nodavirus and EMCV, in that they're both plus-strand RNA viruses.
Right.
Okay,
The
and they
difference-
have to replicate through an intermediate that's going to generate double-stranded RNA.
Correct. The big difference is that no one has identified a viral suppressor of RNAi in EMCV. Whether there is one or not is actually an interesting discussion. Now, they show in one figure that you make canonical phased siRNAs when you infect these embryonic stem cells with EMCV.
Can you define phased for me? I'm confused by this.
Phased is just, imagine dice are going down the genome and chopping it up every base or every couple of bases, right?
Does it mean that does this imply that there's a preference for RNAs that are from a certain region? Okay.
Yeah, well, I think phasing doesn't imply any preference. It just means it goes down the genome and chops as it goes. But there is
overlapping.
Yeah, overlapping.
Okay.
So you've got you might have an siRNA from bases 1 through 22 and bases 2 through 23. And,
yeah, exactly.
and so you've got overlapping pieces.
I see. Okay.
Phased is a strange term to apply here, but that's the way people say it.
Well, what about Figure 3F, where they show these specific read sequences?
Mm-hmm.
That implied to me that
there's a bias for
that there's a bias.
So
there is a bias for certain regions
Right.
in the end, and that's important. But I'm not sure that that's the same as phasing, unless it's a specific type of phasing.
Well, for example, if there's a bias for the ends of the genome, right, the 5' and 3' ends, within those ends, the siRNAs are phased. In other words, you don't just get one siRNA and then one next to it, but they overlap. They're nested, I guess. Is another way to look at it.
In DNA language, they would call those contigs, yes?
Yeah, you could call it a contig.
Mm-hmm.
Yeah.
So I think this is probably what's been established in the field, phased. So, and that's what we got.
Yeah, and that allows them to plot these polar diagrams where they register how the distribution of the pieces goes along the RNA and how it's phased, so to speak.
Right. So it's phasing in any case, but if the phasing register is focused in some place, you can say that the RNAs are coming specifically from that region of the genome.
Right.
Okay.
So the first experiment, they infect these ES cells with EMCV. They show a northern blot for one of the capsid proteins, VP1, which shows you that it increases from 3 to 6 hours, but there's no measure of virus yield, which I think is a big problem because you would like to know. They find siRNAs, but what's the effect on virus multiplication? So they're being made, but the virus is apparently multiplying. So it would have been nice to know what's going on there. A corollary is that they differentiate these cells, so embryonic stem cells can be differentiated, and when they do that, this decreases the pool of siRNAs that are produced from the EMCV genome. Okay,
Right.
So the idea is that as these cells become more somatic-like, this siRNA machinery is now no longer able to process the EMCV genome. Now, if you think about that, it's weird, right? Because the previous paper said that somatic cells could make siRNAs, but maybe there's an inhibitor of siRNA in the genome that's working in somatic cells, but not in
Well, and
undifferentiated?
somatic cells ramp up as they differentiate. They ramp up the interferon response.
So that could be kicking in as well.
Which is a different approach to the same problem. So you're going to use the RNA as a trigger for interferon, which is a protein-based antiviral response. So maybe it's taking over.
So it would be nice to know what the actual virus replication levels are, right? The only thing we have here, we have a few northern blots for VP1, which show as these cells differentiate, the levels of VP1 remain the same. So the differentiation, the decrease in the amount of siRNAs that occurs as you differentiate these cells apparently has no effect on the replication of the virus.
With something as basic as that, Vincent, and that sounds like a basic criticism of the papers, why didn't the reviewers originally suggest that, and then why didn't they put that into this paper?
I mean, maybe the virus doesn't make infectious particles in these cells. I think that's not probably it, because these are mouse cells, and that's what EMCV grows in. But I would have asked for some virus yields here, because if you look at, I hate to call out figures because most people won't be able to look at it, but figure 2E, there's a northern blot in differentiating these cells. So you infected undifferentiated stem cells and then differentiated. Right. And the amount of viral protein is the same. VP1 is the same in both cases. And in fact, if you can knock out dicer in these cells.
Right.
Now, when you knock out dicer, the amount of siRNAs goes way down, but the viral replication isn't affected.
Right.
As measured by VP1. So you're making what look like siRNAs in these embryonic stem cells. They go away when they differentiate. But there's no effect on virus replication. That's weird in my view. Mm-hmm. And that's a criticism I got from the paper.
It's not weird. It just doesn't support their hypothesis.
Right.
Right.
That's another way to say weird, but I would just say the data don't correlate to the conclusions.
Yeah, well, so there's no direct demonstration that these siRNAs that are produced are antiviral.
Exactly.
Right, and that's what's lacking here.
Exactly.
Right. What they measured as the output of virus doesn't seem to be affected by them.
Now, they say, and this is a nice little statement that they make here. Let me find it. Demonstrating antiviral activity of siRNAs entails the genetic rescue of VSR-deficient viruses in RNA-compromised cells, an approach not possible with EMCV because we don't have a VSR, a viral suppressor of RNAi. All right? So this kind of leaves us in a lurch with these experiments because we can't go any farther. So they move to nodavirus, and they infect these embryonic stem cells. They use the differentiated ones, and they show that just like in the other paper, with wild-type virus, you get very little siRNAs produced. You certainly don't get the polarity distribution. And with the deletion of B2, you get lots of the right canonical siRNAs, and the B2 mutant doesn't grow very well in these cells at all, as demonstrated by northern blot.
And once again, at this point, that's a correlation.
That's right.
Okay? It doesn't grow as well. You don't necessarily, at this point, know that deficiency in growth is because of the interference.
Right.
It's a correlation.
Now, what they do is they look at a quadruple Argonaut knockout embryonic stem cell line. So these have all the Argonaut genes, which are used to process to process the viral RNA using the siRNAs as a guide. They're all gone. And they show that the deletion mutant grows much better, as measured again by RNA levels in those cells compared to in wild-type cells. So there's some connection between the RNAi machinery and viral replication.
Right. So an advantage of these ES cells is you can actually knock out the RNAi system either by knocking out dicer or by knocking out the Argonaut genes. And then look and see what effect that has on the virus, which is kind of the experiment that you wanted to see in the other paper, but that wasn't really possible.
Right. So this made me think, now, if in fact, let's say that RNA interference is antiviral in mammalian cells, then all RNA viruses have to have evolved a viral suppressor of RNAi.
Well, they have to have evolved a solution for the problem.
Oh, that's what I mean. A VSR is a solution.
Well, but it doesn't necessarily have to be a specific suppressor. I mean, the virus could have some other strategy for getting around it. Maybe it just replicates so fast the system doesn't kick in.
Well, what immediately comes to mind for me is reovirus that conceals its double-stranded RNA from the cell all through its life cycle by keeping it in capsids. So there are other ways around the problem.
But these picornas, polio, EMCV, all the viruses we work with, they're in the cytoplasm. They're making tons of double-stranded RNA. It's not hidden as far as we could tell.
Well, how about the vesicles in which this stuff is made, right? You get these, what do they, bud in or out from the ER?
So in plants, the RNAs replicate inside the vesicles, and that may be to shield it from the RNAi machinery. But in mammalian cells, the RNA replicates on the surface. So anyway, the point is that no one's identified bona fide or convincing suppressors or ways to get around this in mammalian viral genomes. And I asked Chris why, and he said it's been really difficult because if you have a protein with an RNA binding phenotype that's there to antagonize interferon, that will often read out as a suppressor of RNAi, but it may not really be one.
Right. Because the adult cells have this interferon system that throws a huge wrench in this, which is, I think they point out in a couple of places in these papers and in the review articles, that's been one of the biggest problems with figuring out whether this system is really functional in mammals.
Yeah. The other interesting issue is the paper that Chris talked about last week from his lab, which shows that in mammalian cells, the RNAi machinery, at least in part, is there to suppress the expression of interferon-stimulated genes, many of which are toxic. And that when a cell is infected, then the interferon response, one of its jobs is to turn off the RNAi machinery by poly-ADP ribosylating, I can't remember what was the target, either Drosha or Argonaut.
I think it was Argo.
So it shuts it off. So how can that be? How do you reconcile that with these observations in this paper?
Well, to me, the reconciliation there is that that may be what's going on in differentiated cells. Okay, this RNAi thing, according to at least one of these papers, is probably at least more robust in pluripotent cells.
Perhaps,
okay?
Yeah, perhaps.
Right.
So
and this could be a this in some sense could be an evolutionary relic. We've had this since we diverged from plants, maybe, and it's there. And in cells that, for whatever reason, haven't turned on their interferon response, maybe the system is functional. But once the interferon response has been turned on, this is just sitting there. As I think we've said before, evolution is kind of bureaucratic. It doesn't get rid of any of the old stuff. So this is just sitting there in the background, and maybe in a lot of these viral infections, it's not functional.
I think that's probably a good explanation. These primitive cells, stem cells, don't have an innate response yet, right?
Right.
So they need something to protect them, and we know that stem cells can be infected by viruses, so that maybe that's what this is. And then as they differentiate and the innate system kicks in, which may be more effective, then the RNAi is used for other things.
In which case, viruses that principally infect somatic cells or adult cells aren't really going to need a way to deal with this system because they're not going to encounter it.
Yeah. So that's the question. Can you find suppressors in viruses? And I've been thinking about that a lot because we work on these viruses, and I'm wondering, is this an interesting thing to work on? But it sounds like a lot of people are banging their heads against the wall.
Yeah.
So the conclusion of this cell review is that this result is certainly provocative and should prompt experiments designed to examine the ability of pathogenic viruses to generate siRNAs in vivo.
I think that's an appropriate summary, right? So this is evidence that this thing exists, and we need more experiments to really lock this down and prove the relevance. I think that's fine.
But there are some things that are apparently contradictory that may be resolved at some point that need to be resolved. And I think they need to do plaque assays.
Yeah.
Don't you think, guys?
Yeah.
That's what they should have a picture of instead of the mice.
Yeah.
So anyway, yeah. So these were cool. I tell you, I think RNAi is really interesting, and it's clearly it was just in early days.
Yeah.
Listening to Chris last week, it's so evident. Yes. Ken?
Yeah. I was just going to say I had just a couple of random thoughts. One is that from the picture again in the review that's in Science of what happens in the suckling mice versus the adult mice, just made me start thinking about those viruses for which the pathogenesis is different when they infect newborns versus adults, like LCMV or polyoma or things like that. And then just listening to Chris talk about how he thought things were important in the DNA viruses that can persist, and questions about do RNA viruses have this kind of ability, just made me start thinking about RNA viruses that persist with defective interfering particles, and made me start wondering about if there's some kind of RNA interference mechanism that might be at play there.
So with LCMV, if you infect newborn mice, they're fine throughout their lives, right? They're tolerant to the virus. I think that's the pattern. And if you infect an adult, they make an immune response and they die. So I wonder what's going on. I'm sure someone is looking at the RNAi response there, because that may turn out to be a crucial player. Mm-hmm. Maybe it controls it early on. Dickson, are you okay with this? Do you get this?
I follow everything you've been saying.
Okay, because I want to make sure if we make it clear to you.
Yeah.
That's important.
No. I have a clear picture of what needs to be known and what is known.
Now, what Chris said last week on TWiV was that viruses appear to have co-opted this RNAi. They've got sequences. Some viruses have in their genome sequences that look like RNAis and can be shown to target genes, and that's another growing field. But not every virus. So Chris's idea was that the viruses that tend to persist are the ones that are going to have RNAi sequences to try and allow them to persist.
Viruses that really need to fine-tune gene expression and sort of establish some sort of homeostasis relative to the host need this sort of fine-tuning, as opposed to viruses like, for example, pox, that just go in and slam dunk, the hell with everything.
There's no finesse there, right?
No finesse at all.
So pox has no viral suppressors of RNAi.
Well, it's got it does encode a very robust double-stranded RNA binding protein. Mm-hmm. So if there was an RNAi response, pox has the potential to squash that. But it doesn't encode any of its own microRNAs that we know of. There's also the potential for pox to, it makes double-stranded RNAs late during infection, so there's potential there for substrates for things like dicer, but it's all happening in the cytoplasm, blah, blah, blah. There's no evidence that pox at this point is really messing around with the RNAi system at all.
All right. Some email.
Yep.
Kathy, can you take that first one, please?
Sure. Linda writes, Dear TWiV, I received my PhD in microbiology in 1994, worked for a while, and then somehow was out of the lab for 10 years, taking some time off with my kids and developing and teaching online courses. The kids are older now.
I don't think your kids counts as time off.
No. You switch to another job.
Yeah, that's right.
Exactly.
Right. The kids are older now, and I really wanted to get back into research. I was lucky to find someone willing, or desperate enough, to give me a job in his lab, and I started last month. It is a virology lab, and I really knew nothing about viruses, having only worked with bacteria. So I was trying to find some basic virology information online. I feel like I hit the jackpot when I found TWiV through iTunes. During the first episode I listened to, Vincent mentioned his virology class at Coursera, so I signed up for it. I really have learned a lot about viruses by listening to TWiV and taking the course. Thank you so much for making these materials available. I've also started listening to TWiM and TWIP, and I'm enjoying these as well. I'm still teaching basic microbiology online. Our new term started at the beginning of September, and I've been including information about viruses during each week because I really find them fascinating. Thanks again.
Cool.
Nice.
Cool.
Thank you.
Very nice.
Very cool.
Dickson.
Suzanne writes, the chocolate-dipped candy Dickson mentioned is Pucky. I only know this because my husband's comic book shop used to sell it. His husband has a comic. Sometimes I'll see it in the international section of a large local supermarket. The shop still sells Pokemon. That's a popular and possibly somewhat addictive collectible card game. Sounds like Alan already knows about that one. We've succeeded in keeping it away from our kids so far. Kind of hypocritical of us, I know. On a totally random note, the students in my 10-year-old's class are giving reports on various fields of science and what the scientists do. My daughter picked virology. Mostly, I think, because I mention things from your podcast to her occasionally, or have to explain jokes when I laugh at something you all just said. She came home from school the other day and said excitedly, "Do you know the people who make This Week in Virology also have This Week in Microbiology and This Week in Parasitism? And did you know that blue eggshells are caused by a virus?" I guess she Googled you up on your blog. I don't think she'll end up as a career in science. She says right now that she wants to be a singer. But you are surely doing your part for keeping the rest of the laity interested in science. That's a good thing, too.
I think that's great.
Yeah. The meek shall
10-year-old.
inherit the earth.
10-year-old.
Yeah.
Yeah.
Love it.
And this
Cool.
is an image of the candy?
Yeah, that's one I found. Right.
Well, that's not the
Pocky.
real Pocky.
No.
That's like a homemade version.
Yeah, Pocky is like
The
little
real
thin
ones are
sticks.
very, very uniform.
That's right.
They're phased.
Or contigged, as the word may be. Yeah, right.
All right. Alan.
Matthew.
Sorry. Go ahead.
Okay.
Yeah.
You sure now?
Take it.
Yeah, go ahead. Well, the thing is, Kathy had solicited, but we'll let her take care of that. Yeah, go ahead.
Right. So I'll read the letter and Kathy can answer it. Or I'll read, we'll figure it out. Anyway, Matthew writes, my Spanish isn't good enough to understand this. Links to his site, somosbacteriayvirus.com. I wondered if you'd heard of this guy and what you think of his work. Cheers, Matthew. And Kathy, you looked into this.
Right. So I wrote to Isabel Novella, who was a fellow Holland Lab alum. In fact, she was his last postdoc. She's now at the University of Toledo. She's from Spain, and she's an expert on viral evolution. So I had translated the page using Google Translate, cutting and pasting, and figured out that she would be a reasonable person to answer this. And so she replied, "I've never heard of this dude, but the university where he was teaching is very good. It's the institute where Esteban works. That's part of it. It seems he was unhappy and took early retirement, so this is his personal webpage. I haven't read his papers, and I'm going on the content of the webpage. He claims that neo-Darwinism is BS, and there is no evidence that proves it. He is not anti-evolution. His main beef is that evolution doesn't happen in small steps, but in big jumps. His model establishes bacteria as the main raw material for genes and viruses as the mechanism to produce major changes, either by moving chunks of genetic information or by novel regulation of existing information. I think he is right in that these processes are happening and they are evolutionarily meaningful. He's in the same line of reasoning that claims that symbiosis is an argument against Darwinism because once you become a partner, you are not necessarily the fittest, in quotation marks. My own take, and this is still Isabella, my own take is that neither claim is anti-Darwinian in itself or proof that the neo-Darwinist model is insufficient. Darwin never got into how you generate diversity. He started with the fact that there is diversity. We keep finding more new ways to produce variation. That evolution goes in fits and jumps was proposed by Gould.
That's right.
Darwinian, if there ever was one, several decades ago, the very famous punctuated equilibrium.
Indeed.
At the end of the day, two things matter. A, what works, and B, what, if any, is the competition. The rest is time.
Hear, hear.
The point that I definitely did not like is that he claims that there's no proof or demonstration that Darwinism works, and that is simply false, really. And then she says parenthetically, okay, assuming he doesn't mean literally, because of course you cannot demonstrate anything, only something that is false. But that's true for all hypotheses and theories. Mm-hmm. I hope this works. Cheers, Isabel. And then she points out that on the bottom left of this person's homepage, there he has a thingy where you can choose the language yourself using Google Translate, so you don't have to copy and paste it all out. You can just see it.
I totally missed that, which is why I put it on.
Right.
I feel like an idiot.
Yeah.
I tried to read the Google translation, and it's
rough.
I couldn't. It's rough. Yeah,
Yeah.
it's really tough. I mean, the stuff is heady enough to start with, so
Right.
that problems with the translation don't help.
Yeah,
Right.
but this doesn't seem like anything really. I mean, the bits that he has any support for don't seem like anything new.
Can someone define neo-Darwinism for me? I'm putting you on the spot there.
Dickson.
Dickson, because I'm a neo-anything.
What's
I guess.
neo-Darwinism?
Well, it's after Darwin, so it means that after genetics was discovered and Mendel's papers were recovered by Dobzhansky and then brought to light, and then Morgan and Sturtevant and Stiles and Dobzhansky invented genetics at Columbia University, by the way, I might add. And then Darwinism started to look different because now we had some genetics to talk about, and instead of just, yes,
okay.
things vary, but how do they vary? So that's when Stephen Jay Gould started to deviate from the classical approach to describing slow, small steps of organisms changing as the environment selected for those changes. And, over a long period of time, you could get new species. And Stephen Jay Gould was convinced because of his experience with the Burgess shales. I know this is a long explanation, but this is what I've been told. He saw that all of a sudden there was this explosion of life about 600 million years ago, and decided that maybe there were some cataclysmic events which could speed up the process of speciation that didn't have anything to do with the way Darwin originally proposed it, and Wallace, too, I shouldn't forget him as well. So neo-Darwinism is this rethinking of the way Darwin originally couched the process of speciation into more modern terminology. That's basically
All right.
it.
Right.
Okay.
Okay?
Right. It's Darwin plus DNA.
Yeah.
Darwin plus
Right.
DNA and viruses. I mean, anything
Yeah.
that generates diversity is fair game for discussing how life goes from one form to another.
Right. So Darwin got the outlines of the process right.
That's right.
And everything we've found since then to try and falsify that has in fact underscored how correct he was.
Exactly.
For somebody who didn't know anything about DNA or
That's
how
right.
viruses work.
That's right. That's
It's really kind of shocking. But
Yeah.
this punctuated equilibrium concept, which if you read Origin of Species, there are
Right.
some bits and pieces in it in which
Yeah.
Darwin kind of hints even at that. But the idea is it's not, when you look at the fossil record, a very, very gradual change of forms over time. What you see is things are stable, and they're stable often for a very, very long time. And then something happens that destabilizes that ecological equilibrium,
Yeah.
and everything evolves very rapidly for a period and then stabilizes at a new in a new status.
Sure.
But certainly viruses provide evidence that you can have slow selection, right? Not jumping in evolution.
Yeah, but
right?
maybe
They
one
do
little...
both.
They do both, sure.
Of
Yeah.
course they do. And one little change can result in some very remarkable rearrangements.
Yes.
So...
Well,
you know,
it depends. I mean, if you select a drug resistance in a virus, a single amino acid change, so he would probably not define that as a big jump in evolution, right? Because you're not changing species or something like
No,
that.
but if that change wiped out an entire genera...
Yeah, paleontologists tend to look at gross phenotypes rather than genotypes.
Yeah, that's
yeah,
right.
yeah.
And the gross phenotype can change radically in response to
course.
a very small genetic change.
Yep.
That's right. And the other thing that is never considered, because there is no record of it, is the introduction of infectious diseases into the mix. I'll give you the best example, and that's the island of Hawaii. When the Culex mosquito was introduced, nothing happened. When the Culex mosquito with malaria was introduced, that changed the landscape entirely. 5 genera of birds were completely eliminated from those islands as a result. 5 genera. That's an enormous number of animals. And in their place, others arose to fill in those niches so that the ecological setting didn't change, but the species did. So that was huge and very, very fast. So there.
Anyway, thank you, Isabel. That was good.
Yeah.
Rich Condit, can you read Roberts?
Sure. If I can, a little distracted here. Here we go. Hello, TWiV hosts. I just finished listening to episode 247 with Dr. Lipkin. When the show ended, I immediately went to the EID paper and read the article. Paragraph 7 of the article contains the following, quote, a product, nucleic acid, from a fecal pellet of T. perforatus bat showed 100% nucleotide identity, end quote. In the webcast, between 11:15 and 11:20, Dr. Lipkin says that the sample was a fecal swab, not a fecal pellet. I remember that we had some discussion over this. This exchange was prompted by Dr. Racaniello's question about the nature of the sample. So was the sequence obtained from a rectal swab or a fecal pellet? I guess I'm nitpicking here, and I'm not sure it makes any real difference to the importance of the finding. Let me just stop at that point and say I would trust the paper, not the investigator.
Not the memory of the guy who wrote it.
Yeah, I trust what's written down rather than the PI.
Yeah, no, you're right on that one.
Okay?
Right on that one.
I am a retired biochemist immunologist, and after 3 years of my being, this is good, after 3 years of my being around the house all day, my wife suggested that I find something to do.
Get out of my hair.
I didn't think I was doing anything, but then after 30 years of my being gone most of the day at work, I will grant that I may have disrupted her routine. So, being an academic at heart, I decided to revisit calculus at my local JC. That wouldn't have been my first choice.
No, me neither.
That was 6 years ago, and I'm now 6 classes away from a BS in applied mathematics.
Oh, my God.
This semester, I'm taking a breadth course, quote, principles of gene manipulation, end quote. We've had 2 lectures, and at least 10 minutes of each lecture have been taken up with questions that relate purely to vocabulary issues.
Of course.
I think the authors of Principles of Virology are definitely on the right track with striving to avoid the use of jargon. I hadn't thought much about this until now, and I agree with Kathy that proteins are expressed on the cell membrane. I've asked a few of my friends what they think, and we all come back to proteins are expressed on the cell surface. I suppose we could say that proteins are incorporated into the cell membrane, but old ideas die a hard death. I found TWiV when I started taking the Coursera virology class. Love the class and the lectures, and so does my wife. Well, we've come full circle there. That's good. Looking forward to eventually listening to all of the TWiV episodes and the next issue. Weather in Orange County, California, was wonderful. Clear skies, 77F. 25C, 2.52, humidity 47. Winds 5 to 10 miles south-southwest. Great day for a sail, but alas, class instead.
Yeah.
Robert, P.S. I have a copy of Dickson's book on West Nile fever, and I was wondering if he would sign it for me.
Of course I would.
There you go.
How does that work? You can send it to me, and I'll send it right back to you if you'd like.
Probably. PSS. Rich mentioned in one of the episodes something about the time frame for unraveling the actual DNA code. I would recommend anyone interested in the history of the discovery of the DNA code to read the following, and it's Leder and Nuremberg from 64, Code Words and Protein Synthesis 3 on the nucleotide sequence of DNA coding units. I was just in the process of trying to look up that particular paper, and I believe that that's the one where they actually used the ultimate solution of all of the code words involved taking synthetic triplet codons and in a tRNA binding assay asking in a complex with ribosomes and tRNA which triplet codon bound which tRNA. And it was putting together that assay that was critical in figuring out ultimately the whole code. And that 1964 paper and that date is where that's how I mark sort of the birth of the genetic code. There was a lot of stuff that, before and after, but that to me was the founding paper.
Right.
And it's a great paper. That's really a landmark paper. I also had a very red underlined and annotated copy of 1966, the genetic code 31 Cold Spring Harbor Symposium. This volume contains a fantastic number of papers by the founders of molecular biology, lots of great pictures, too. Any large university or medical school library should have a copy or two. Yes, back in the 60s and 70s, I don't know about so much now, but the Cold Spring Harbor Symposium each year picked a current topic like the genetic code, or a couple of years later, 69 was protein synthesis. And these volumes, they got together all of the major players and who gave sort of talks that were published in the volume. And they're incredible resources for the papers of the day. And they had a photographer at Cold Spring Harbor who went around and took pictures of all the participants that are in the front of the volume. So you get to put faces with the players. So they are really great reads.
Right.
It's fun to look at those now and see some of the people when they were young.
Absolutely.
I have a pile of them I inherited from my colleague, Dave Figursky, and it's just great to look at the players. So, Dickson, is this why you come in here every day, to get out of your wife's hair?
No, she goes her way and I go mine. She goes off to art class and I come in here to become a better person. Which I hope is working.
Yeah, you just,
so that's a great letter and great recommendation.
It's very good.
Yes.
Yeah, absolutely.
Next one is from Jackie. Dear doctors of TWiV and valued guests, I have written previously but have not written recently so that I can give myself a chance to get caught up on the back episodes. Up to 207 now. However, there are a couple of things that I don't think could wait any longer. The first is, due to the government shutdown, the PubMed-saved search engine is sending out nonsense emails rather than my weekly update on what is new in my small realm of science. So I wondered what else people use to keep up on the most recent articles in their fields that are not government-run. The second is, I know that TWiV tries to stay out of political issues, but I wondered if you could make an exception in this case and get someone on to talk about the ramifications this shutdown might have on the scientific machine, especially related to the NIH, but also to other areas of the government. Now for the part that could have waited, but that I am going to talk about anyway, since I'm already writing to you. I have, as I said, been catching up on back episodes, and a number of them of late, 25 episodes before 207, have discussed the evolutionary arms race between viruses and hosts and the Red Queen hypothesis. I've wondered for a long time about the advent of modern medicine and what effect it might have on the evolution of our species. Modern medicine is quite young in evolutionary terms, and evolution is very much about characteristics that will keep the population as a whole propagating. Modern medicine is very much about individuals surviving and propagating. Now, I am pro-modern medicine, but I sometimes wonder about the broader implications this might have on our species in the long term. I wondered if the TWiV team would be able to comment on this. The second thing I've wanted to write about and haven't is a listener pick of the week. I've been using StumbleUpon now for several months and have found amazing sites that I would never have come across without it. Basically, it's a website that you can subscribe to and then tell your interests, and then you push the Stumble button, and the site takes you to a random site in that area of interest. You can then let StumbleUpon know if you like the site, dislike the site, or want to save the site to your favorites. It then tries to learn what you enjoy so it can give more of whatever that is. Warning, this can be a giant time sink if you like this kind of thing, but I find it very entertaining and sometimes educational. Jackie is in the People's Lab at the Research Institute at Nationwide Children's Hospital.
Cool. People's is the PI's name. It's not the People's Lab, like the People's Car.
Or People's Park.
Yeah. Or the
Where
People's
is
Republic.
Where is the People's PEOPLE's lab? Who knows? We've talked about this issue of medicine interfering with evolution on TWiM a little bit. We have here, you and I, Dickson, have talked about
David Attenborough even came out and said, for people, evolution has stopped because we now can interfere with what would ordinarily have been a lethal situation. We can keep them alive and allow those genomes to continue to replicate.
But I don't think evolution has stopped.
He was trying to be dramatic. He's a television naturalist, but he's a good naturalist. And I know what he meant, and I know how he said it. But it's interesting to think about it in terms of that, and we've changed it. Let's just put it this way. We just changed it.
Yeah, so people who would have died from infectious diseases of various sorts and
Or
whose
metabolic
genes would be
disorders.
removed.
Yeah, like
Exactly. So they're here,
That's
and the outcome is it's going to be different from without medicines. It's clear.
It's true. But that's only for the people who have access to modern medicine, and that's a small fraction of the total Earth's
That is a
population.
very small minority of the human
Very,
gene
s- even in the United States. I mean, in a developed country like this, with all the issues associated with getting to modern health care, no politics intended, of course, it's not an easy deal.
So what is the population currently of the globe, Dickson?
7.2 billion.
All right. So how many of those have access to medicines?
Less than 1%.
Well, which medicines? If you mean the full spectrum of modern medicine, I think, yeah, probably about 1%.
So very little effect on evolution then,
Very.
at least from medicines. But there are other things.
They all
that are-
use medicine. Everybody out there uses something to cure something with. And who knows who's got life-saving drugs available through traditional approaches. And in fact, Pharmacia would love to know the answer to that question.
Well, the answer that they've found so far is probably not much.
Yeah, I think you're right. The average lifespan, though, if you go to a country like Cuba, which has been hamstrung with regards to their availability of modern medical interventions, they have the longest-lived women's population on Earth. They're over 85 years on average. So that's an interesting foil for the concept of if you don't have access to modern medicine, you're going to die early. That's not
Well,
true.
but they do have access to some of the more crucial parts of
Yeah,
modern
no,
medicine.
that's right. I don't
Their
say-
their public health infrastructure in Cuba is actually pretty impressive for what they have to work with.
That's not considered modern medicine. I've taught at a school of public health all my life, and trust me.
Oh, no, I understand that.
I mean, it should be included, but it isn't.
From my perspective, the greatest innovation in modern medicine has got to be the flush toilet.
Yeah, that's right. Or that's
Indoor
true.
plumbing
No, you're
trumps antibiotics.
Totally
or,
correct.
And if
Yeah,
we're looking for a
you're
second
right.
place, I'd have to say window screens.
So when you have your
No,
fatal,
living in northern climates.
Alan, when you have your fatal gastroenteritis, you can sit on a toilet rather than a hole. in the ground.
Exactly. Yes. The things.
You won't be
things-
sharing it. You won't be sharing it.
The things that kill people at young ages
Yeah,
and
that's
have
right.
the biggest impact on lifespan and reproductive success are the things that we've cured with mostly very simple technologies and interventions.
I agree. And then living in a northern climate. I was told forever that the best cure for a tropical disease is to live in the
Live
temperature.
in a northern climate.
Try window screens, right?
Yeah, that's true too.
So how can you measure, though, the effect on human evolution? I don't think we can do that at this moment, right? We just think that it's happening.
Yeah.
But because of the access problem, it can't really be happening yet.
No. It's certainly not on a grand scale that would affect the outcome of the human condition.
Yeah.
That's right.
And also, what are we looking for the impact of this? I mean, if the human gene pool changes as a result of modern medicine at a point when everybody has access to modern medicine, who cares?
Right.
If that somehow makes us less able to survive in a world that lacks modern medicine, but our world has it universally, then there's no impact.
Yeah, the biggest step in human evolution is yet to occur, and that will occur when we learn to live somewhere else besides Earth.
Yeah, we may go somewhere and there's no medicine, or maybe a
Well,
war
there will
happens
be.
and
No,
it wipes out our modern medicine.
Our
right?
microbiomes will change dramatically.
Then there'd be a punctuated equilibrium.
That's right.
Exactly.
Okay.
Maybe Stephen G. Gold had something going on.
Back to one of Jackie's early questions about how do you keep up with the literature if PubMed search engine is down. I've, for a long time, gotten things from the ASM journal alerts. There's an e-table of contents that you can get, and also something called Site Track, where you can customize what you want. And you don't have to be a member of ASM, it looks like. You just have to give them an email address.
Right.
And there's a pretty wide range of journals that you choose from. It's not just the ASM journals. And so that's been pretty good for me.
So I use RSS feeds of each journal anyway, and that is independent for most journals of the government shutdown, which now is over, of course.
Yes.
So you can go to every journal's website. There's usually an RSS feed, and then you can put it into a reader. I use Feedly, which is a browser-based reader, and then you can have all of your journals and just scan through them quickly.
Very cool.
All right.
I want
Yes,
to go
sir.
back just for a second. I've been distracted with this genetic code thing.
Okay.
Oh, hi, Rich.
You can get wrapped up in that,
Right, the article that Robert cited was one of the earlier Nuremberg and Leader articles that uses techniques to figure out a couple of codons. There's a later article in 1965 that capitalizes on the triplet binding assay, which was developed in an earlier paper, and basically, I believe, solves the rest of the codons, and I pasted in a link to that there. I'm thinking of an article that has a simpler presentation that I'll bet you was in the 1966 Cold Spring Harbor Symposium paper that has a very simple table that's the results of all of it. But for people who are interested in this kind of stuff, those are just classic papers. So I had to get that off my chest. Thank you.
All right. Hope you feel better.
I do.
All right, let's do one more email. Kathy.
This is Becca, right? Yes. Greetings. I'm a high school Spanish teacher, and I've been listening to your podcasts during my commutes to work since 2011. TWiM, then TWiV, then TWIP, each as I started running out of another, working my way from the first episodes to the present. At first I was listening in 1.5 speed, eager to catch up. Now I'm kicking myself because I'm out of episodes by Tuesday each week. It could be Wednesday if TWIP weren't TMIP, meaning
This is a real junkie.
I think this means this month in parasitism would be TMIP.
Well, that's why we dribble them out, come on.
I decided to look for some other podcasts to supplement the TWiVs. I remembered the sites I've heard about, Science Podcasters, for example, but found none of those sites that have gone under. Sorry. She's found that those sites have gone under due to lack of time or funding. I tried listening to Mundo de Microbios, which has a lot of good information, but I found that it just doesn't simulate the conversations I found us having in the car. I am thus cordially looking for some advice on good podcasts to listen to from my lovely hosts or any other listeners. To broaden the search, I can even listen in Spanish. I find that at the end of a long day with high schoolers, I need to have some adult interaction, and strangely enough, that need is usually satiated by listening to your podcast.
Cool.
Any ideas?
No, repeat them. Go back and
enough.
listen to them again and slow them down, and you'll have a wonderful time.
Right?
Well, I don't. Can I interject, Kathy?
Sure.
I don't think there's anything like the TWiVs, to be honest. But if you go over to Science 360, the NSF site.
Radiolab might be another.
No.
Hey, I was on Radiolab. It's okay.
Science360.gov. The NSF actually collects all the science podcasts that are out there. So it's a one-stop shopping, if you will. I would recommend, it's not conversational, but Mark Crislip has a few podcasts. The Puscast is one of them, which is about infectious diseases that you might like. So check those out. Okay.
Also, wasn't there somebody who we had at least briefly on a show who was going to do a Spanish Esta Semana en Virología or something like that?
That never got off the ground.
Oh, never happened. Oh, that's too bad.
Mañana.
There is the epidemiology public health podcast out of Johns Hopkins that we're supposed to be guests on at some point, but I don't know I haven't found the link for that, so I don't know how it goes. I haven't listened to it. But check out science360.gov. Radiolab Dickson is a radio show. It's not a podcast. It's highly produced. They put millions of dollars into it.
I'm aware of that. I was on the show. I know this.
But
it's entertaining.
It's not conversation.
It's entertaining.
It's way overproduced.
It's entertaining.
You should be ashamed of yourself.
Not that there would be anything wrong with somebody putting millions of dollars into TWiV.
No, there wouldn't be anything wrong with that whatsoever.
If they did,
we would just keep doing the same.
We
wouldn't
same thing.
Change.
I wouldn't overproduce it. I would just
hair
get some help.
on our lack of hair's heads.
What would we do with all that money?
Travel.
So whenever someone invited me, I would say I'm bringing my whole crew and paying for them.
Yeah.
Right? That's what I would
fly
do.
out wherever we're going.
Anywhere, and I'd bring a tech person who would set up. And
we do on the site interviews and in-depth reporting.
And I would just do it every week.
And I would be your sidekick.
Hey, this week we're going to Austin. Who wants to come?
That's right.
Every week, and then while we're there, we could interview local people, do 2-hour interviews, and stack them up and release them. We could do so much.
Vincent, why did you make fun of this guy Icon? He could actually support us if he's listening.
Who said I made fun of Icon?
You mentioned something about him. So let's repeat
Icon,
that.
You're talking about Carl Icon?
Yeah.
No, he comes in and takes stuff over and changes it.
Yeah, see, we don't want that. We don't want that.
That's his business.
No, we don't want that. No.
All right, let's finish that one up.
Yeah, Becca continues. You can read this on whichever of the TWiVs you choose. If you read it on TWiP, I'd like to make a strange inquiry. Vincent suggested that using some sort of cat urine barrier to keep out mice. Wouldn't this just keep out the non-infected mice?
Of course.
Aren't you thus inviting the infected ones to stay in your house as they wouldn't be scared off? Or perhaps any non-infected mice in your house would be afraid to leave. Curious if this was just my naive misunderstanding.
No, you got it right. You got it
No,
right.
she did, yeah.
She's got it right, nailed it. So you
Yeah.
just can't play with Mother Nature like that, Vincent, and expect good things to happen.
I think that this
scientists
is,
always get it wrong.
I get it wrong. That's not, other scientists are fine. I'm really impressed that this is a Spanish teacher in high
That's
school.
remarkable. That's
Yes.
remarkable.
Because I look at my kids' teachers, all of them, and
Yeah.
I say, these people are never going to listen to TWiV. And I just
know,
don't know why. But I think this is great
Yeah,
that you're
me
listening
too,
and that
me
this
too.
is not your field.
I have a great.
And she put together this really good
Yeah.
idea.
Let me insert some anecdote here. When I was in high school, I took Spanish, and of course I did miserable at it because I wasn't very serious as a student in high school. And my teacher's name was Mrs. Di Nazario. She was remarkable. She was highly tolerant of intolerance. And she was from Puerto Rico, and she was a wonderful, kind person. And I actually registered for Spanish 2, and she made me promise that if I did, that I wouldn't register for Spanish 3, which I didn't. So at the end of my 4 years at Dumont High School in northern New Jersey, she signed my yearbook, and she signed it thusly. Dickson, your smile will take you far. It took you through Spanish 2. Love, Mrs. Di Nazario. So we had a wonderful personal relationship, and I never studied, and I felt so disappointed at the end. I had such empathy and sympathy for her after I entered the teaching field and said, how could I have been so stupid and insensitive as a small child? And, most of us have had similar kinds of experiences, and here she is. This is a struggling woman that needs adult content for her world to become more holistic. And I'm so pleased that we can supply this. So I'm now giving back to Mrs. Di Nazario the things that I wish I could have, but couldn't. So please accept my remarks as such.
Nice.
Yeah.
Very nice.
Deep sympathy.
Let's do some picks of the week.
Okie doke.
Alan, what do you have for us?
I have a serious pick for a change of pace from my usual. But last week, the science journalism community had a bit of soul searching about sexual harassment, and it led into a whole discussion about women in science and the problems they face. And there were a whole bunch of very insightful and thoughtful posts and discussions on Twitter. If you want to search on Twitter, the hashtag to look for is # and then ripples of doubt. That'll give you dredge up a whole long discussion in 140 characters, and it'll also link you to a lot of the relevant longer blog posts. But one of the ones that I just found really, really both depressing and heartening that this is being said outright was this blog post by Hope Yaren, and it's titled "The Worst Part Is Not," and it's a sort of a brief history of her own experiences as a scientist, and from the perspective of a woman coming up through the tenure track. And I was just really blown away by this. And I think everybody in science should probably read it. And I also ran this past Kathy before picking it, because it's one of these. If you're what I saw in my Twitter stream was women who read this said yep, and men who read it said, oh my God. So it's just I think it really starkly highlights the radically different experiences that women and men have of the way things operate in the scientific field. And Kathy, I'll let you take it from there.
Alan wrote to me, and I found it compelling. I think that almost every woman in science can relate to you something that happened that might have been more minor, might have been more major, or she knows of someone to whom things happened. So, we know from the sociology of science articles and things that people say, well, it's getting better, isn't it? Isn't everything okay now? And yes, it's getting better, but no, everything's not okay now. And we had that in I think it was your pick, Vincent, a couple of weeks ago. A woman writer from University of Michigan who is an English professor who had been a physics undergraduate, and then pursuing that topic, she visited the Yale physics department and thought that things would be all better now, and in fact found that some things hadn't changed much at all. So there are a lot of enlightened men out there, and this kind of thing probably helps to enlighten both men and women about the fact that there are still some barriers.
Well, this is from a lab of my colleague here at Columbia, which I didn't know. All right, thank you, Alan. Rich Condit, what do you have?
Right. I got a serious one, too. Man, this is really serious. I was reading that plot. That's really something. Okay, my pick is a book called The Universe in a Single Atom by the Dalai Lama. And this is the Dalai Lama is kind of an amateur engineer scientist. He's always had an interest in science and technology from a very young age. And he wrote this book that kind of explores the subtitle is The Convergence of Science and Spirituality. So he explores the convergence or overlap or abutment of science and spirituality. I think it's important to distinguish. He draws a very clear distinction between spirituality and religion. I think not everybody has his definition of spirituality in mind. He's defined spirituality as concerned with qualities of the human spirit, such as love and compassion, patience, tolerance, forgiveness, contentment, a sense of responsibility, a sense of harmony. So, he's actually on record as saying spirituality you have to deal with, religion we don't have to deal with. Okay, that's a different issue. Okay. And just to give you some idea, I'm going to read a little thing here from his prologue in this book that gives you an idea where he's coming from that I think is very interesting. It says, I remember a disturbing conversation I had only a few years ago with an American lady who was married to a Tibetan. Having heard of my interest in science and my active engagement in dialogue with sciences, she warned me of the danger science poses to the survival of Buddhism. She told me that history attests to the fact that science is the killer of religion, and advised me that it was not wise for the Dalai Lama to pursue friendships with those who represent this profession. By taking this personal journey into science, I suppose I've stuck my neck out. My confidence in venturing into science lies in my basic belief that as in science, so in Buddhism, understanding the nature of reality is pursued by means of critical investigation. If scientific analysis were conclusively to demonstrate certain claims in Buddhism to be false, then we must accept the findings of science and abandon those claims.
Wow,
that's pretty strong stuff. And the book is the book is very interesting. And the most interesting part is that the Buddhists really are very interested in the nature of consciousness, which can be approached by just sitting on your butt and looking at what goes on in your head, or by sticking your head in a functional MRI. And so there are two very different approaches to understanding the same thing, and he looks at that and other things as well. I have a sort of a sub-pick, which is a link to the Mind and Life Institute, where he has regular meetings where he invites scientists to India to participate in conferences where they explore these same issues.
Have you been there, Rich?
I have not, but I think it's on my bucket list.
Cool.
The Beatles have.
Yeah?
Yeah.
All right, thank you, Rich. My pick is a Nature news article that happens to be open, so you can get it. It's not behind a paywall. It's called The Truth About T-Rex. I always liked dinosaurs. As a kid, I still do. And this is just a cool article about all the stuff we'd like to know about T-Rex, It turns out we have a lot of skeletons of T-Rex.
We do.
Really lots. So there's a lot of things that people can investigate, like whether it was covered with fuzz or scales.
Or feathers.
And what it looked like as a youth. Ute.
Yep. A ute.
Do you know what movie that's from?
I do.
Yeah, that's My Cousin Vinny.
Exactly.
Great movie.
Fabulous.
One of the best.
Fabulous movie.
Ute. What did you say? Okay. Whether it's feathers and how it got its prey and so forth. It's a cool article anyway. So
I like this figure in it. It's
very nice.
Yeah.
Dixon, what do you have?
I have the winners of the current Yahoo Nature Photography Contest. And it's just the visuals. And it's just, there is so much peace of mind that one can garner from a single snap moment of time in nature, where nature is behaving like nature. And that's what they were looking for, and that's what they found. And these are wonderful pictures, and I just like to sit in front of them sometimes and look.
They are absolutely gorgeous.
They are.
There's one with the crocodiles on the mother's head. That's so
Yeah.
cool.
The captions are neat too.
They are.
Number 9 is the Japanese macaque around
Nice.
the hot spring in the snow.
Yeah.
That's pretty cool.
This is life being life, and that's what we're all about
Nature.
here anyway. So
Nature's pretty cool, Dixon.
very, very cool. Very
Oh,
cool.
they have a lot of interesting... Oh, look, they have a slideshow from Sandy last year. Wow.
Yeah, well, that's
Nice.
nature at its rawest.
I'm going to send this right off to my family.
Here, here.
That's very cool.
And I'm going to buy that book for my son, by the way, Rich. That's
Thank
a great
you, Dixon. I appreciate
book.
it.
That's quite all right. It's a pleasure being part of this group.
Kathy, what do you have?
Well, I have a pic that's John Holland on YouTube. So for those of you that didn't ever get to go to one of his classes,
Okay, good.
UCSD put this on. He gave this lecture as an emeritus professor in 1996, and it's about emerging infections. It's almost an hour-long lecture. So if you want to just hit the highlights to get the flavor of John Holland, I would
Cool.
say see the first minute or two, and then you could skip to somewhere around 14 minutes where he starts talking about how roadkill is rendered into animal feed. And then
I listened to that. It's
Really?
amazing.
then I would
Really?
say go to 48 minutes to the end, and you'll get it all. It just had some real laugh out loud spots for me.
Wow.
And the roadkill into animal feed had a connection this morning on the news. I heard about an emerging story in China from Chinese pet jerky.
Yeah, that's right.
Is killing animals. 600
Pets.
pets so far,
That's right.
and 3,600.
It's probably melamine. It's got melamine in it, I'm sure.
Yeah. So
It's tragic. It's tragic.
and I think the pet deaths are here in the United States.
They are. That's exactly right.
So
It's crazy.
anyway,
Crazy.
I digress. But anyway, check out John Holland.
Kathy, did you know that there's a chain of restaurants in New England called Roadkill Diner?
No.
Oh, yes. And you can drive into them, and they've got wonderful iterations of common food items couched in terms of roadkill. So, yeah,
It's nice that there's a video of John. That's
It
good.
is, yeah.
roadkill.
All right. We had a listener pick earlier. I have one more from Stephen, who writes, the International Institute for Species Exploration site does not relate directly to viruses. If viruses aren't alive, how can they have species?
Oh, dear.
On the other hand, it's eminently suitable as a science site pick, and it's beautiful to boot. Credit for my discovery goes to Michael Novacek, Museum Provost of Science at the American Museum of Natural History, who cited this site in a Science Cafe talk on the AMNH podcast. Provides a link to the podcast and a link to the home. Finally, here's a choice from the top 10 new species of 2013 that Vincent will appreciate, at least for its social media connection. And it is a picture of a lacewing
Oh, good.
with
It's a pest control animal.
a spider on its back.
It's in the Neuroptera.
In a trend-setting collision of science and social media, Hawk Ping Gook photographed a beautiful green lacewing with dark markings at the base of its wings in a park near Kuala Lumpur and shared his photo on Flickr.
Right.
Dr. Sean Winterton, an entomologist, serendipitously saw the image and recognized the insect as unusual. When Gook was able to collect the specimen, it was sent to Dr. Brooks, which confirmed it as a new species.
Fantastic.
So there you go. Social media, new species. That's
Right.
cool. Thank you, Stephen.
Terrific.
You can find TWiV at iTunes and at twiv.tv. And if you like what we do, go over to iTunes and leave a rating or a comment. That helps us to stay visible. And do send us your comments and questions to TWiV at twiv.tv. Dickson Despommier can be found at verticalfarm.com, medicalecology.com, and Racaniello.org, and also at the Huffington Post.
Here, here.
Thank you,
Oh.
Dickson.
Yeah, it just got accepted. So
I'm not sure that's a good thing.
it's not an obligatory every week type of
I know. I'm just
blogging.
kidding.
And no, it's a good thing.
The Post tends to steal stuff from other
Yeah, I
news
appreciate
sources.
that too. I appreciate that.
Not
too.
Dickson.
But
Yes,
not
sir.
Dickson. They got the
No,
original.
damn right.
Thank you, Dickson.
Someone already stole me.
Rich Condit is at the University of Florida, Gainesville. Thank you, Rich.
Sure enough, great time.
Kathy Spindler is at the University of Michigan. Thank you,
Thank-
Kathy.
Thanks. This is a lot of fun.
Go blue.
Go Blue, yeah.
Go blue.
Alan Dove is at turbidplaque.com and also on Twitter. Thank you, Alan.
Always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.
God, what an adventure! Where have we been in the last hour? We've been everywhere.
A lot of
this
fun.
is incredible.
You know what, Richard? I totally agree with you. This is like,
I mean, I come out of this and I go, where am I? Where have I been?
that's right.
It's incredible.
It's transcendental viral meditation.
The show is
cool.
totally immersive.
It is cool.
It's fantastic. It's great.
Ah.
My amygdala thanks everyone here.